Yes, good morning, dear students. So, we have finished off with the concept of hormones, and today we go into a small but very crucial topic, everything about cell division. School में थोड़ा थोड़ा किया है. There are two types of cell division, mitosis and meiosis. Here, of course, we go into tremendous detailing how exactly mitosis takes place and how exactly meiosis takes place. So let's understand this beautiful topic of cell division. Cell division states that there is a cell theory. Every cell arises from a pre existing cell. And this is achieved through the process of cell division. The life of all multicellular organism starts as a single cell called as the zygote, and the zygote undergoes a series of continuous cell division, helping in growth and development of every living organism by cell division. And you have something called as the cell cycle. Cell division is a process by which a cell divides into two or four daughter cells. If it is mitosis, in mitosis takes place once, and say 44XY becomes 44XY and 44XY. So chromosome number remains the same and cell division takes place once. But in case of meiosis, if you remember, 44XY undergoes meiosis 1 to form 22X and 22Y, and that is called as meiosis 1. And 22X then becomes 22X and 22X, and 22Y becomes 22Y and becomes 22Y. So, sir, you have four cells formed, and the second division was called as meiosis 2. So, in case of mitosis, one cell divides to form two cells. In case of meiosis, one cell divides to form a total of four cells. And in mitosis, chromosome number remains the same. 44XY becomes 44XY, 44XY. In meiosis, 44XY becomes 22X, 22Y. So chromosome number becomes half, and two cells divide to form four cells. So a diploid cell forms two diploid cells, that is in mitosis. In meiosis, a diploid cell forms two haploid cells, and two haploid cells form a total of four haploid cells, which is meiosis 2. This we had done when we had done reproduction. Here we are doing it in more detail. Cell division is a process by which a cell divides into two or four daughter cells, two during mitosis and four during meiosis. Now, a cell divides when it has grown to a certain maximum size. Daughter cells formed after division of the mother cell grow in size and divide again. So, sir, ये है mother cell and it will divide to form daughter cells. And daughter cells will grow in size. And after they grow in size, again divide to form daughter cells. So, cell division becomes a continuous process, and continuously you have one cell becoming two cells becoming four cells will divide to form eight cells, and eight cells will divide to form 16 cells, and so on. And that is the way continuous cell division will take place. So 1 becomes 2, 2 becomes 4, 4 becomes 8, and so on. The period between two successive divisions is called as the generation time. दो सेल डिवीजन के बीच का जो टाइम होता है is called as the generation time. A series of changes or sequential events occurring in the life of a dividing cell is called as cell cycle. तो एक सेल से दूसरा सेल का जो टाइम होता है is called as the generating time or generation time. And all the changes that take place in a cell for forming daughter cells is called as cell cycle. तो एक सेल साइकिल के बाद you will have daughter cells form, and that series of changes or sequential events occurring in the life of a dividing cell is called a cell cycle. A cell cycle includes the period of beginning of one cell division to the beginning of the next cell division. एक सेल डिवीजन से दूसरे सेल डिवीजन का जो टाइम पीरियड होता है is the cell cycle, and the processes which take place within a dividing cell is called as the generation time. And a cell cycle consists of two main phases: interphase and division, or the M, mitotic or meiotic phase. So chahe mitosis ho ya meiosis ho, there are two main phases, namely the interphase and the division, or the M phase, which is mitotic or the meiotic phase. So what exactly is the interphase? The interphase was formerly called the resting phase, but the cell is highly active and prepares itself for the following cell division. Hence, it is now called as the preparatory phase. So, interphase, which was initially called resting phase, is now called as the preparatory phase. It involves many metabolic activities, and the interphase is further divided into three phases: G1 phase, S phase, and G2 phase. So एक सेल ने अभी अभी दो डॉटर सेल बनाए हैं. Each daughter cell will undergo G1, S, G2. Each daughter cell will undergo G1, S, G2, and then will divide to form two more cells. So ये कौन सा फेज था? इंटरफेस. और इंटरफेस में क्या क्या स्टेजेस हुए? इंटरफेस में हुए G1, S, and G2, which are the preparatory phases. And what are these phases? G1 phase is the first growth phase, which is also called as the post-mitotic gap phase. So, samjho a cell has divided to form two daughter cells. The cytoplasmic volume has become half because one cell has become two daughter cells. Cytoplasm आधा आधा हो गया है. In the G1 phase, the cells will grow in size. The daughter cell will grow in size and reach the size of the mother cell. And that is called first growth phase, also called post-mitotic gap phase. It involves synthesis of proteins, enzymes are formed, and energy molecules, ATPs, are formed. So during the growth phase called G1 phase, you have protein synthesis in the ribosomes of the rough endoplasmic reticulum. Protein synthesis takes place, and in the mitochondria, ATPs are formed, which are required for DNA replication, and proliferation of cell organelles take place. You have more number of Golgi bodies, more number of peroxisome, more number of chloroplast, mitochondria, ये सब proliferate होते हैं. The cell may withdraw from the cell cycle and enter into G0 or the quiescent state. तो सर एक सेल G1 फेज से S phase में जा सकता है, or may go into G0 phase. And From the G0 stage, it may re-enter the G1 phase and again undergo cell division, or may get differentiated and become a permanent cell. तो सर एक सेल जब G1 फेज में जाता है, G1 से इट गोज इनटू G0 फेज, एंड G0 फेज से इट कंटिन्यूज इन द G1 फेज or may undergo stopping of cell division. It remains locked in the G0 phase, and that cell is called as a differentiated cell. So, example, you have meristematic cells. Meristematic cells are undergoing continuous cell divisions, and meristematic cells are dividing continuously. A meristematic cell will undergo G1, S, and G2 phase, and then undergo cell division. But a meristematic cell may go into the G1 phase, go into G0 phase, and stop cell division and may get differentiated to become parenchyma or sclerenchyma or collenchyma, and cell loses its power of cell division. तो parenchyma, collenchyma, sclerenchyma बन जाता है and loses its power of cell division. So what is a differentiated cell? A cell in a G0 phase is undergone a differentiation, and a loss of control at the G0 phase leads to uncontrolled cell division as in tumor or cancer. एक सेल G1 से G0 फेज में जाता है और G0 फेज से it does not enter a normal G1 phase, but it goes into an uncontrolled cell division phase, and it has lost its power of cell division, which is an uncontrolled division, and that is called as a tumor or a cancer. तो G0 फेज से वो डिफरेंशिएट होकर परमानेंट बन सकता है, या उसका लॉस ऑफ कंट्रोल हो सकता है G0 फेज में, जिससे एक कैंसर या ट्यूमर बना सकता है. That is the G1 phase. So you have G1 phase, and that is a growth and normal metabolic activities of a cell cycle, the G1 phase. From G1 phase, the cell may go into S phase. S phase is also called as the synthesis phase, in which primarily the DNA will replicate. तो डीएनए रेप्लिकेशन होता है. एस फेज इज द फेज ऑफ सिंथेसिस और सिंथेटिक फेज. Replication of the DNA takes place in this stage, and the entire genetic material, the DNA duplicates. So DNA का content double हो जाता है. The nucleus also doubles in size, and histone proteins required for the organization of chromosomes are also produced. RF sir must have just taught you DNA अकेला नहीं होता है. Along with the DNA, you have the DNA. And here I'm just showing you the DNA. This is your DNA. DNA is never present as a double thread. The DNA is wound around specific proteins which are called as histone proteins. And around the histone proteins, you have DNA thread. And that thread is actually called as a chromatin fiber. So here we have DNA, then we have histone protein. We have DNA, we have histone protein. We have DNA, we have histone protein. And histone proteins are around which the DNA coils. And that fiber is called as a chromatin fiber. तो स्कूल में ये सुना है, न्यूक्लियस हैज इन इट डीएनए और chromatin thread, chromatin fiber. What is chromatin fiber? It is DNA plus the proteins called as histone proteins. So histone proteins also multiply in the S phase. S phase में histone proteins also multiply. तो S phase में histone proteins also multiply. So DNA multiplies and histone proteins multiply. Along with the histone proteins, the centrioles duplicate. तो तीन चीजें होती है. सेंट्रियोल बनता है. DNA, histone and centrioles. Now what are centrioles? सर ये है सेल, ये है न्यूक्लियस, और न्यूक्लियस के बाजू में एक छोटा सा एरिया होता है which is called as a centriole. Centriole बनता है and there is a small cytoplasm around it which is called as, ये cytoplasm होता है which is called as centrosome. ये centrosome होता है and this is the cell and here is the nucleus. And during the S phase centrioles duplicate, तो एक सेंट्रियोल की जगह दूसरा सेंट्रियोल विद सेंट्रोसोम आ जाता है एंड बोथ सेंट्रियोल्स गो टू द ऑपोजिट पोल्स and both centrioles go to opposite poles. In the future, between the centrioles start getting arranged spindle fibers. Spindle fibers कहां से कहां बनते हैं? From one centriole to another centriole, spindle fibers are arranged. So spindle fibers are arranged from centriole to centriole. Spindle fibers are arranged from centriole to centriole. So when does centriole duplicate? Centriole duplicates during the S phase. So S phase में तीन चीजें मल्टीप्लाई होती है. The DNA replicates, histone proteins replicate, and the centrioles replicate. याद रखना एस फेज सबसे इंपॉर्टेंट है. डीएनए, हिस्टोन प्रोटीन्स एंड सेंट्रियोल्स रेप्लिकेट ड्यूरिंग द एस फेज. एंड देन फ्रॉम एस फेज द सेल गोज फ्रॉम सिंथेसिस फेज into second growth phase. Second growth phase is growth and preparation for mitosis. First was growth and normal metabolic roles. Formation of enzymes and ATPs were there. Enzymes and ATPs were there. यहां पर भी ग्रोथ होता है एंड प्रिपरेशंस फॉर माइटोसिस होते हैं. ये था G1 फेज, ये है G2 फेज. तो growth takes place in G1 also, growth takes place in G2 also. So once again, a cell has divided to form two daughter cells. Both daughter cells undergo G1 phase. G1 phase में cell will grow. Inside it, enzymes, proteins are produced. Inside it, ATPs are produced. And that is called G1 phase. After G1 phase, S phase. एस फेज में ये है G1 फेज. फिर एस फेज में डीएनए रेप्लिकेट होता है, हिस्टोन प्रोटीन्स रेप्लिकेट होते हैं एंड सेंट्रियोल्स रेप्लिकेट होते हैं. सो दिस विल हैपन इन द एस फेज. एंड G2 फेज में ग्रोथ एंड प्रिपरेशन फॉर माइटोसिस होता है. एंड विच प्रिपरेशन फॉर माइटोसिस? एक प्रोटीन बनता है कॉल्ड एज ट्यूब्यूलिन. तो ट्यूब्यूलिन नाम का एक प्रोटीन बनता है. ट्यूब्यूलिन इन द फ्यूचर फॉर्म स्पिंडल फाइबर्स. याद है इन सेल डिविजन सेंट्रियोल्स के बीच में स्पिंडल फाइबर्स अरेंज हो गए. स्पिंडल फाइबर्स आर मेड ऑफ अ प्रोटीन कॉल्ड एज ट्यूब्यूलिन which is also called as actomyosin, which Vishal sir has just taught you. Actin and myosin. Actin and myosin का actomyosin होता है. And what is the property of actin and myosin? Can undergo contraction and relaxation. So similarly, spindle fibers, which are made of tubulin or actomyosin, also undergo contraction and relaxation. When is the tubulin protein formed? During the G2 phase. G2 phase में tubulin. तो once again G1 में क्या होता है? ग्रोथ. एंड ग्रोथ के साथ एंजाइम्स और एटीपीज बनते हैं. एस फेज में क्या क्या बनता है? डीएनए रेप्लिकेट होता है, हिस्टोन्स रेप्लिकेट होते हैं और सेंट्रियोल्स रेप्लिकेट होते हैं. एंड G2 फेज में क्या होता है? ट्यूबुलिन दैट इज एक्टोमायोसिन इज फॉर्म एंड सेल अगेन अंडरगोज ग्रोथ इन साइज. तो G1 में ग्रोथ, G2 में ग्रोथ, एस फेज में डीएनए, सेंट्रियोल्स एंड हिस्टोन्स अंडरगो रेप्लिकेशन. G1 में enzymes and ATPs are formed. G2 में tubulin, actomyosin, which is required for formation of spindle fibers is formed. And that is G2 phase. It is the second growth phase, also called pre-mitotic gap phase or growth phase. It involves the formation of tubulin, the spindle protein, and RNA. Also, there is duplication of cell organelles such as mitochondria and chloroplast. Number of mitochondria and chloroplast will increase. And आपके notes में दिया है centrioles duplicate and two centrosomes are formed. Now, this actually is taking place in the S phase. तो यहाँ centrioles duplicate and two centrosomes are formed. ये please cut कर देना. Where is centrioles duplicating in the S phase. And the cell now is ready to enter the mitotic phase. So, sir, G1, S and G2 phase, ये तीनों phase खत्म हो जाते हैं. Cell is ready to enter mitosis. What is G1, S and G2 phase? Collectively called as G1, S and G2 is collectively called as the interphase. Now, from the interphase, the cell will go into division phase. Division can be in mitosis or meiosis. तो सर दो टाइप के ग्रोथ पॉसिबल है. टाइप्स ऑफ सेल डिविजन, although all cells divide, the division is atypical. Binary fission in prokaryotes. बैक्टीरिया में एक सिंपल डिवीजन होता है which is called binary fission. And binary fission से one bacteria will divide to form two bacteria. So that is a very simple primitive type of cell division called binary fission in bacteria like prokaryotes, which is atypical. And it is typical in eukaryotes where the cell division may be of the following type. You have amitosis, mitosis and meiosis. So actually चार टाइप के सेल डिवीजन देखे हमने. Binary fission in case of prokaryotes bacteria, which is atypical, and typical in eukaryotes, multicellular organism, in which you have amitosis, mitosis, and meiosis. Let's do amitosis, mitosis, and meiosis. Today we will focus on the type of cell division called as mitosis. इसके ऊपर focus करते हैं.